So welcome back. Now we will see the last step of replication, which is called as the termination and primer removal. So first we will see primer removal. So primer removal happens in both the lagging and leading strand. So here when we see both the strands, the leading as well as the lagging strands, so here you have the template here, which was the template strand, which was 3' to 5', and now we had our replicated strand, which was 5', and the newly synthesized strand, which is here. So this is this one is the leading strand, or the LES. So here what happens is, after the complete synthesis, the primer is removed by, in the case of bacteria, it is by pole 1. It removes, because this is an RNA, it is not a DNA, it is an RNA, this will be removed. This primer will be removed by the DNA pole 1. As we said earlier, pole 1 is having exonuclease activity. With the exonuclease activity, it will remove this RNA, and because of its polymerase activity, it will add the DNA, it will replace it with the DNA molecule. And now there is no problem with the case of the leading strand, because the leading strand will be perfect, the length of the strands will be perfect, that of the template. But the problem arises in the case of the lagging strand. Now I am going to take the lagging strand alone. So in the case of the lagging strand, the lagging strand has had an orientation which is so this was the template for the lagging strand, and we saw it was synthesized in many pieces, which was called as the Okazaki fragment. This is a 3' end, and this is the 5' end. So what happens is, after the Okazaki fragments are completely synthesized, these primers, all these primers, this is a primer 1, this is a primer 2, and this is a primer 3. These primers will be removed, or it will be removed by our DNA pole 1, as done for the leading strand. It will be removed by the DNA pole 1 and will be replaced with RNA. As the same thing happens here, our RNA pole 1 is having only unidirectional polymerase. It can synthesize only in the 5', 3' direction. This particular primer, the synthesis of this particular primer will be halted. This primer synthesis, that is this particular the synthesis of after the removal of this last primer. So when that particular last primer is removed, I will take our strand will be like this. This is a 3' end, this particular strand 3' end, and this will be the 5' end. Here we had a primer. When that primer is removed, now this is a 5' end. This extra portion, that is this extra portion, cannot be synthesized again by our polymerase, because polymerase can synthesize only when there is a free 3' end. Because here you are having a free 3' end, this particular portion of the DNA will not be replicated. As a result, after the synthesis of the Okazaki fragment, after the synthesis of the complete Okazaki fragment, it will be like this. Our synthesis after the synthesis, that means there will be a gap, or there will be a small portion which was left unsynthesized. This is what is called as the end replication problem, and this end replication problem is solved by a particular enzyme which is called as telomerase, which is present in the gametes, the stem cells, as well as in the tumor cells. What the telomerase does is, the telomerase will extend this particular lacking segments in the lagging strand. These particular region should be extended. So extending this particular regions is done by this particular enzyme, which is called as a telomerase. So telomerase, what it does is, it will extend this particular region, in the sense that extending this particular region the end regions of a linear chromosome. We are talking about eukaryotes. This is called as telomeres. Telomeres usually does not the end region of the DNA. Usually, they does not contain the important genes in this particular region, and this telomere is usually elongated. Telomere is elongated by the telomerase. So telomerase is an enzyme. If consider this as the enzyme which is called as the telomerase enzyme, this particular enzyme, inside it is having an RNA. So this telomerase is a reverse transcriptase enzyme. Reverse transcriptase in the sense that it is having an RNA inside. It is going to synthesize a DNA. From this RNA, it is going to synthesize a DNA, which is a reverse of normal transcription process, and hence this enzyme is a reverse transcriptase. So this particular enzyme will synthesize the missing fragments, which is complementary to the RNA. Thus, it will what does is it will extend the telomeric region, extending the telomeric region. And when the telomeric regions are extended, what happens is, even though in the lagging strand, when this particular thing when a portion is being not synthesized or removed, there will not be a problem, because this the fragment which is synthesized by the telomerase is just repeats. It does not have any genes. As a result, ആ ഭാഗം പോയാലും it is not affecting any genes. അപ്പോൾ ഓരോ റെപ്ലിക്കേഷൻ കഴിയുമ്പോഴും ഇതുപോലെ നീട്ടിക്കൊടുക്കും, അടുത്ത റെപ്ലിക്കേഷൻ കഴിയുമ്പോൾ വീണ്ടും ചെറുതാവും. അപ്പോൾ ചെറുതായാലും വലിയ പ്രശ്നങ്ങൾ ഉണ്ടാവില്ല, അതിനൊന്നും നീട്ടിക്കൊടുക്കും. പക്ഷേ ആ റീജിയനിൽ ജീൻസ് ഇല്ലാത്തത് കൊണ്ട് ഇറ്റ് ഡസ് നോട്ട് ഹാവ് ഓർ ഇറ്റ് ഡസ് നോട്ട് പോസ് എനി problem in this particular region because of this telomerase. It is not affecting any genes. So this is the termination process that is happening in the case of eukaryotes. അപ്പോൾ ടെർമിനേഷന് മുമ്പ് നമ്മൾ പ്രൈമർ റിമൂവലും പറഞ്ഞു. പ്രൈമർ റിമൂവൽ നടത്തുന്നത് പോൾ 1 ആണ്. ലീഡിംഗ് സ്ട്രാൻഡിൽ ദർ ഇസ് നോ പ്രോബ്ലം, ബട്ട് ഇൻ ദ ലാഗിംഗ് സ്ട്രാൻഡ് ദ പ്രോബ്ലം ഇസ് ദ ലാസ്റ്റ് പ്രൈമർ ആഫ്റ്റർ ഇറ്റ് ഇസ് റിമൂവ്ഡ് ഇറ്റ് cannot be extended, and that extending the last primer is done by our telomerase. So telomerase is an enzyme. Telomerase is an enzyme which cannot copy the അതിന് മുകളിലുള്ള കോംപ്ലിമെൻ്ററി ഡി എൻ എനെ. അപ്പോൾ ഇവിടെ ടീലോമറേസും വന്ന് മൈൻഡ് ചെയ്യുമ്പോൾ ഇറ്റ് കനോട്ട് ടേക്ക് കോംപ്ലിമെൻ്ററി ഫ്രം ദിസ്, ബിക്കോസ് ടീലോമറേസ് യൂസസ് ഇറ്റ്സ് ഓൺ RNA ഫോർ സിന്തസിസ്. അതുകൊണ്ടാണ് നമ്മൾ നേരത്തെ പറഞ്ഞത് അതിൻ്റെ RNA വെച്ചിട്ട് തന്നെയാണ് എക്സ്റ്റെൻഡ് ചെയ്ത് പോവുക. അപ്പോൾ ഇത്രയുമാണ് ടെർമിനേഷൻ. അപ്പോൾ ഈ പറയുന്ന ടീലോമെറിക് റീജിയൻ, അതായത് ഇൻ ദ കേസ് ഓഫ് യൂകാരിയോട്സ്, ബിക്കോസ് ഇറ്റ് ഈസ് എ ലീനിയർ ക്രോമസോം, റെപ്ലിക്കേഷൻ ബബിൾസ് അറ്റത്തെത്തുമ്പോഴേക്കും ഇറ്റ് ടെർമിനേറ്റ്സ് ബൈ ഇറ്റ്സ് ഓൺ. അവിടെ പ്രോട്ടീനോ ടെസ്റ്റ് പ്രോട്ടീൻസോ ഒന്നുമില്ല. ഇറ്റ് is being arrested, or it is being dissolved, or the enzymes will move out from the DNA by its own when it reaches the ends. So that's with the last portion of replication, which is termination. Okay, and now coming to the difference between prokaryotic and eukaryotic replication. അപ്പോൾ ഈ ഡിഫറൻസസ് നോക്കുക, നമ്മൾ പറഞ്ഞ കാര്യങ്ങൾ തന്നെയാണ്. അപ്പോൾ ഒരു എസ് ചോദിച്ചാൽ, ഇഫ് ഇറ്റ് ഇസ് എ 10 marks question, യു ക്യാൻ ആഡ് ദിസ് അറ്റ് ദി എൻഡ്. അപ്പോൾ നമുക്കിവിടെ ഡിഫറൻസസ് എന്ന് പറയാൻ only you have this much differences. Comparing eukaryotic and prokaryotic replication, the process is the same. That is how a leading strand is synthesized, or the initiation process, elongation, termination. We saw the difference because it is linear. We said the difference in termination, but the other one, that is initiation, elongation, and the primer removal, etc. All of the three processes remain same for eukaryotes and prokaryotes, except the enzymes involved are different. The mechanism is the same, but the enzymes will be different. That's the only difference in eukaryotes and prokaryotes. The other difference is regarding the termination, and also some minor differences which is listed here. So with this, we come to the end of this particular session, which is DNA replication. So there will be doubts, because this is little complex in regarding the lagging strand synthesis, etc. So whatever be the doubts, you can either post in WhatsApp or in the Google Classroom. So we'll discuss. If you want further explanations, further clarifications in this, just put me a message in WhatsApp or the Google Classroom. So thanks for listening. So we'll come up with the next session. Next session will be probably transcription or translation. Bye.